Hello, this is the second delivery of our project, which will include the methodology for overexpression of the Micromonas pusilla NIA gene. The first part is the extraction of agal RNA, which includes the RT-PCR for the NIA gene and the first electrophoresis. The second part is the enhancer selection, plant DNA extraction, restriction digestion, the second electrophoresis, and the gel extraction and PCR. The third part is the ligation of the gene and promoter, then the vector digestion, the third electrophoresis, and the second gel extraction and PCR. The fifth part is the ligation of the insert and vector, and then the 3RE comparison method. And the last part is the transformation of yeast cells, their growth, and the colony selection.
RNA extraction, we will do a cell lysis and then use the 3 reagent, that is a vanadium threosinate phenyl chloroform, to do a separation where the RNA remains on the aqueous phase, as we can see here, and then we use a precipitation with isopropanol so that our RNA stays here. Then we proceed to do our reverse transcriptase, where we will be using our reverse transcriptase as the method to convert our RNA into a complementary cDNA. So this then will be followed by a normal PCR that has its four main parts. That is the initiation, the denaturation, where the bonds of hydrogen are broken, then the annealing step, where the primers are annealed where they have to be so that the elongation can start, and the complementary sequences and wanted sequences will be then amplified.
Then comes an electrophoresis for verification of the procedure. RNA stain for viewing the bands is used. In the first well goes an RNA ladder, in the second one the total RNA as control sample. Then there is the RT-PCR product in the next 3 wells. A 1.6 kilobase band is expected, being the NIA gene. One research team found an enhancer-like promoter of the NIA gene in Arabidopsis thaliana called NIA1. It induces the translation of nitrate reductase and causes a 4-time overexpression. This is the promoter we chose to use. DNA from the plant is extracted with the CTAV method. This detergent disrupts the cell membrane and a high salt concentration precipitates polysaccharides. The plant is first put into CTAV and SDS and incubated, before washing with isomyl alcohol, isopropanol, and ethanol 70% in order, with certifications in between them. It is resuspended in TE buffer and purified with cesium chloride.
The next step is the restriction digestion. According to the protocol of Wang et al. In 2010, for the obtaining of the whole NIA1 fragment, they use the enzymes HIN3 and PST1 to achieve the overhangs in the promoter. These enzymes work at 37 degrees Celsius, so at this temperature is going to be incubated and digested. It's also necessary the use of buffers O and P from thermofissures to guarantee optimal conditions and stability. To verify the correct digestion, a second electrophoresis takes place. In this case, after the high DNA ladder, a control will be added, algae DNA sample, and the following wells are going to have the digested DNA. The expected results are going to be 2 bands, and the one near to the total DNA size, and the other near to the last ladder. This means it's going to be the promoter. The size of this ME promoter is 1.8 kilobases. For the gel extraction, we are going to take the small band, melting the gel, precipitate it with isopropanol, and adding ethanol with the respective centrifugations. For the purification, we're going to use the TRIS buffer, and for the amplification, we will use a normal PCR process with the master mix kit, conducting the 35 cycles as we can see in the table below over here.
A ligation will then be done for joining promoter NIA1 with the gene NIA. The promoter has 5' HIN3 and 3' PST1 ends. The gene has its sequence and the poly A as ends. Personalized adapters from Lubio Science are used for their ligation, along with the needed buffers and the T4 DNA ligase, all done in the thermocycler. The shown G vector was selected for its integration in Saccharomyces cerevisiae. It contains an ampicillin resistance gene, which will help as a selectable marker. This vector expresses E2 crimson, but this gene will be cut and replaced with our insert. Restriction enzymes EcoR1 and XBA1 cut E2 crimson cleanly out of the vector, leaving space for the NIA1 NIA insert. The incubation temperature will be of 37 degrees Celsius. using the tango buffer for the double digestion.
For the third electrophoresis, this will help us to prove the vector digestion. We will use newly agarose gel and viewing the bands in SYBR safe DNA stain. The ladder for this electrophoresis will be the master ruler DNA ladder mix from Thermo Fisher. The control will be the undigested sample from Glick Lab, and the last 3 wells will contain the digested vector. The expected results will be 2 bands, one near to the total vector size and the other one far below, being the E2 crimson. In this case, it's important to keep the big band for the extraction. And finally, we will follow the same gel extraction and PCR processes as the ones previously mentioned in this presentation.
The insert has 5' Hint 3 and poly A ends. The vector has 5' EcoR1 and NextBay1 ends. Adapters added in the PCR were used for the correct ligation to happen. The ligation is done in the thermocycler with the T4DNA ligase buffer.
We will do then a 3 restriction enzyme comparison so that we can verify the orientation and the correct vector obtention. We will use XPOL and A-Curry. Then we will use a third restriction enzyme that will cut in the middle of our cutted fragment so that we can see if our vector was done and modified as we wanted to. Here we have how our enzymes cut, and we have that expected bands of 2.4 kilobases and 1 kilobase obtained from our 3 cuts.
Now I'm going to explain the last part of the methodology, which is the transformation of yeast cells to insert a specific plasmid into a specific G strain in order to recover and analyze a limited number of transformants. For this part, a YPAD medium will be used to inoculate and incubate the yeast strain. Then, boiling the carrier DNA to denature it and pelleting the cells in a microcentrifuge. After that, we're going to be adding transformation mix, which will help with the resuspension of the pellet. And after incubating in a water bath, the transformation mix has to be removed with a micropipetter. And with the help of the microcentrifugation, so that the pellet can be stirred and the cells resuspended, to then pipette the samples onto SC selection medium plates and incubate them. The yeast needs to be grown under certain conditions in different Petri dishes. One is going to be the normal yeast cells in SC media as a control. The second one is going to be the transformed yeast cells in the SC media. The third one is going to be a normal yeast cell in the SC media with ampicillin. And the last one is going to be the transformed yeast cells in SC media with ampicillin. And for the selection of colonies, it is expected that only the yeast cells that have the recombinant vector will grow in the SC media with ampicillin, and those cells will be used for further multiplication. And these are our reference. Thank you for watching.